All right, here we are back again, and I wanted to show this again because I showed it at the start of class last time. This is not the distribution for our grade, but a student asked me yesterday about this. It's for a different course, just to show you that all the courses I teach, I do this as a grading strategy. It's intro bio. Our grade distribution is at the start of the answer key video on our Brightspace page. And so what should you do this week? Well, go through that answer key video. I explained why in the previous one. We've used those for the quizzes, and it's considered an essential part of our course. And we're going through the cell cycle chapter now, chapter 18. Well, actually, we're going through this chapter 17, and we'll get to chapter 18 today. But chapter 18 is really split in two: programmed cell death and stem cells. And we're dealing with the stem cells with a homework assignment that's due next Tuesday. And our exam will be on Thursday after spring break, and cover that material, okay? And so we have a nice package of what happens with the DNA damage checkpoint at the start of M. So the G2 to M transition has that DNA damage checkpoint. We've got CDC25 phosphatase, we've got CHEK1, we've got CDK1, we saw cyclin B, we've got the inhibitory phosphates like that. And then what did it do? Once it gets activated, well, it gets helper kinases going, Aurora and Polo. And then we have two examples of targets: histones to condense the chromosomes, to help condense the chromosomes. They also phosphorylate other chromatin proteins like cohesin and quintensin that all work together to condense the chromosomes. And then we have the nuclear lamina getting phosphorylated. So it really shows the biochemistry and the reality of just what happens, what does that mean, how is it a switch, and then what does it mean when it's stopped, and what does it mean when it goes, and what does it mean when it's a cell and it's M. And we saw that the big rate-limiting step in entry into M is we got to remove those inhibitory phosphates. The big exit step from M is we're going to destroy cyclin B. So nice story, nice package for us to start getting a handle on the biochemistry of the cell cycle. Who has these? Every eukaryote and a cell on our planet has that DNA damage checkpoint at the G2 to M transition, and they all have orthologs of all the proteins that I listed. It's really astonishing because we're talking about yeast that causes bread to rise as being an organism that was used to understand the human cell cycle like that, and every other cell cycle of eukaryotes on our planet, right? So then this phases of mitosis is dealt with on the assignment we had due about mitosis and meiosis and the phases of mitosis. That was due on Tuesday. So we've already gotten that part of the chapter, and I just want to show you that it's an important part of the chapter. So you got to be careful when we have an assignment, and there's a video. That material is just as important as what's happening in class. Sometimes it's more important. I'm like, I got to put this together as a package like that. So make sure you study that carefully. And, I'm always trying to guide you toward, well, what do you need to know? And so that includes that, because the book has lots of information in this section, and we want to get pieces of it especially, but then there's additional information. So, for example, you have to know those, and you have to know something that happens, like a kind of hallmark feature of each of those. And that's a really easy question to ask on an exam. Like, what is the most important thing that happens during prophase? Oh, those chromosomes condense, right? Why do they condense? Oh, cyclin B and CDK1 are working together as MPF to just phosphorylate lots of targets like that. So going through there, recognize that mitosis is dealing with the chromosomes, and cytokinesis is a different story. So CDK is not involved in cytokinesis. CDK is active in the role in mitosis, and CDK is doing that job. And cytokinesis overlaps even into some of anaphase, so as the chromosomes are getting dealt with, the cell starts to divide. And our book talks about that, and there's some information in there that we haven't talked about in our class. But here's the contractile ring, and it's made up of actin microfilaments that are going to form a kind of lasso around the big cell and split it in two. So you really want to look through that video and then also go into the book and find the same information there, and also spread out into the book some in that section, because there'll be a question or maybe two from that on the test. for example, this. This is the spindle, and we said we're not going to worry about the spindle assembly checkpoint other than the fact that it causes problems in cancer, and a lot of cancer cells are aneuploid. But with regard to the spindle assembly itself, you know, you should know that those are microtubules that are guiding that, and that we have this type of organization to it. So that's, you know, part of the textbook. That'd be a figure from the textbook, a section from the textbook that we might have a question on the exam about. That would not be something we talked about in class or in that assignment. And so we're especially interested in the molecular mechanisms like we focused on. Now we're talking about no, we're not talking about that. That was phases of mitosis. Another piece about this is really to deeply understand that mitotic index is an indirect measure of start. And that's what it's all about, start. How frequently do cells get to go through start, to go through the cell cycle, and the mitotic index is an indication of that. I just want to show you this because the book has this, and this is a more modern instrument called a flow cytometer. And in a flow cytometer, the cells have to go through it one cell at a time, and it measures the amount of DNA in their cells. And so most of the time cells are in G1, and you see that here. We have a lot of cells in G1, and those cells, based on the video handout for the cell cycle assignment we did on Tuesday, those can be called diploid cells or 2N cells. Diploid means 2 copies of the genome, one from mom, one from dad. And then we're going to go through S phase, and you can see this must be a population of cells that are like skin cells, where they do have a mitotic index and they do have cells rolling through the cell cycle. So in this case, in addition to seeing cells that are in M phase, we also get to see the cells that are in S phase, because it's looking at the DNA content of every cell, and some cells have more than 2N, because they are starting to copy their DNA. And so that's why we see this population in S. And then here, when cells are in G2 and in the first part of mitosis, as they divide, they actually have 4N, because in S phase, mom's genome got duplicated, dad's genome got duplicated. We go from a diploid cell up to a tetraploid cell, a 4N cell. And so just to show you that, because that again is something in the book that we didn't show, didn't really, plan to show in class, but it actually helps strengthen our understanding of what's happening, right? Where, when you're looking under a microscope, you can just see interphase and mitosis. But here, with this flow cytometer instrument, it's looking at the amount of DNA in every cell. And so it finds most of the cells are in G1, some are in S, some are in G2, and they've had their chromosomes copied, and they're rolling into M phase, like that. So it's a really hallmark distinctive feature that the cells in G2 have twice as much DNA as the cells in G1. And the cells in G2 that are copied, have the chromosomes copied, those chromosomes that get copied produce what are called sister chromatids, like photocopies are made during S phase. So, in diving into the book is good, because even the stuff we don't talk about complements the stuff we do talk about. So this is where we're spending our time today and getting through it, and it's the regulation of start. And so we really like this image to just take home and carry with you. There's a direct biochemical pathway between extracellular growth factors like PDGF under a scab and the activation of the cell cycle by activating the G1 to S transition, activating start, turning the light green, letting the cells go into S phase. Because if they get to go through that, they're going to go all the way around and stop at start again. Okay, so that's what we're focused on. And so we know this one in detail. And we can, for the first exam, we took it this far. We mentioned MYC. Now we can take it another step further, because we know in some cells, the rate limiting step is you don't have enough cyclin. And so you need to make more cyclin. So MYC or SRF, somebody's got to be bound to the cis-regulatory region of cyclin and activate its expression. And just one time in our course, we said, lots of cis-regulatory regions are pretty complicated because they have multiple enhancers, right? And so something you should be able to draw on exam 2 is the cis-regulatory region of cyclin, because it's got ELK and SRF on one enhancer. It's got June phos on another enhancer. It's got TCF and beta-catenin on another enhancer. And we learned about all those. And they're used in different settings and different contexts. Sometimes as a checks and balance where it's like, we need both of these to go forward. But other times it's just a different story in a different tissue type, like that, okay? And some of those are enhancers, some of those are activators, some of those are repressors, you know? So it becomes a complicated thing, but it's important because it's a complicated issue. If cyclins are rate limiting, we want to be really careful about making cyclins. Make them if it's the right time, you know, and place, but don't make them otherwise. And so most cis regulatory regions have some complexity to them. Even the myogenin one has more than just the MyoD enhancer. But you know, we got to keep things simple so you can build that framework foundation of understanding, oh, it's a cis regulatory region. And just really once or maybe twice, but I think just once in our course, we're like, oh, actually the cis regulatory region of cyclins is a lot of stuff, okay? So we want to just dwell on that a little bit. And here's just something, AI overview, just to take a look at. So MYC acts as a transcription factor activating genes involved in cell cycle progression, metabolism, protein synthesis. So that just tells you, you know, we're really focused on MYC in the cell cycle. But MYC has a bigger role in that. Think about it, you know, if a skin cell is going to heal, yeah, you got to drive it through the cell cycle. We need some more skin cells, but we should also crank up the metabolism of these guys. Normally, you know, our skin cells are hanging out and doing the kind of the day-to-day work, but under a scab, you know, the skin cells should be, oh my God, I got to step it up, right? And you can think about it because there's a lot of natural selection evolutionary pressure to heal, right? So you're some old ancestral cave character and you got a big old owie on your elbow, you know, you better get that taken care of, right? And healed and have that stiff scab fall out because, you know, you got to get out and hunt and gather, right? Like that, right? So it's just really a satisfying example because it fits together in every way. So it just goes through it, right? Does MYC have lots of target genes? Oh, yeah, it's got a bunch of target genes and other transcription factors are helping and stuff like that. So we're not oversimplifying it. We're not telling any falsehood, but we're trying to, you know, identify some examples on our course that can really show you, oh, okay, I see the kind of steps that are involved, and there's probably just more of them, you know, checks and balances and, you know, activating and repressing like that. So again, MYC, lots of target genes, including these cyclins and cyclin-regulated proteins like that. Then I won't mention that right now because it's a little bit out of place. Oh, what I put it here for is that in this image, we can just crank down, you know, PGF receptor, RAS, all the way down to ELK, and then to MYC, and then to cyclin, and then to this, right? That the cyclin binds to the start CDKs, and they start phosphorylated targets like MCM, which is this mini chromosome maintenance. So we just take it the whole way biochemically from the, oh, you got an owie? I guess you're releasing clotting factor from the platelets and making a scab, and those platelets are also releasing PDGF and stimulating the skin cells under the scab to get going. And just do all the way to that, all the way to saying, let's start working on these origins of replication and start copying the DNA like that, yep. So, you know, really satisfying biochemistry, and it's done that way in every cell, every eukaryotic cell, like that. So, again, you know, we took this a long way for exam 1, and then we could take it further in all the way to MCM, like that, for this one, you know, and you go like, okay, you know, and it's not oversimplification, but it's a really stripped down view of how there is a, you know, direct effect, kind of dominoes falling, series of steps that got to take place. And they vary, you know, and they, you know, need checks and balances and stuff, all right? So there are other stories about activating the cell cycle start. We're just learning a little bit about them, some of them like that, right? So as an example, you know, we talked a little bit about, you know, ERK and SRF, and then we said last time, you know, another target for ERK and ALK and SRF is this phos. So in some cases, June phos is what's going to be a big regular downstream, maybe not so much MYC like that, okay? And so June and Phos come up. They come up in chapter 19 because they're big time oncogenes, because they're kind of, you know, I don't want to say they're related to MYC, they're not related, but they do the same kind of work. You know, they're like transcription factors that say, hey, we better go in through the start, and so we need to activate gene expression. If you had to guess what are the most important targets for June and Phos, you'd say, I bet it's cyclins. You know, I bet it's more examples where cyclins are rate limiting. It's got to be something. They are going to try to make the cell go through start, so they got to affect whatever is rate limiting. Yep. Okay. So there's June and Phos, and you know it's showing there like we drew it there on the enhancer for the cyclins that are active in start. Just a quick aside, remember when we talk about the G2 to M transition and that DNA damage checkpoint, it's cyclin B, right? Do we expect June and Phos to bind to the cis regulatory of cyclin B? No, because they're not involved in that DNA damage checkpoint. They're involved in saying, we got growth that needs to get done, so let's go through start, and let's be bound to the cis regulatory regions of, again, we don't need to learn the letters, but not cyclin B, the other cyclin. Yep. All right. So I just want to show you June Phos. So we just got, you know, we kind of dove into MYC, and June Phos are another big story that we're not going to dive into very much. And here's this other one where we also said, yeah, you know, beta-catenin, that can also activate MYC and cyclins in some settings and be the thing that pushes the cell through the cell cycle. So we also drew the beta-catenin and it's overcome the repression of TCF. We drew that in that cis regulatory region of cyclins. Again, you know, it's got multiple enhancers in different settings to regulate it. All right? So we could do that, right? We could draw a cyclin gene showing enhancer binding sites in the cis regulatory region for MYC, ELK, SRF, June Phos, TCF, beta-catenin, right? It'd just be a drawing. You're just guaranteed to have to draw that on the second exam or the final and label it with all the parts. Our complicated regulatory region. Ultimately, cis regulatory regions are kind of like a set of people voting where majority rules. So even though the story in the cis regulatory region can be complicated, the bottom line is always, are we going to transcribe the gene or not? And if so, how much? Like that. So that's why I say it's kind of a majority rules, because we got a lot of different things that could be happening. What is happening? And is it going to be overall a repression or an activation? Like that. And that's good to keep in mind when studying, you know, transcription factors, is all they ever do is regulate transcription, where, when, and how much. That's it, right? So when things get complicated, you can once in a while settle down and say, well, is the gene getting expressed or not, right? Yeah, and so so under every scab, our favorite example is phosphorylated ELK activated PDGF pathway, right? We got that. We're so solid on that. Phosphorylated ELK binds to the cis regulatory region of MYC and cyclins. It binds to other things too, but that's our favorite. You know, to get that down on an exam and get that down really strongly in your head. All right? So that's our fave, okay? And we got that. We showed this earlier for the first exam, you know, that is MYC required for wound healing? Yes. MYC is activated in skin scales during wound healing, playing a crucial role in the proliferative phase by promoting cell division and migration, right? Yeah, you know, rapid tissue regeneration, rapid healing. The skin scab falls off, you're back in action out there hunting and gathering, right? Like that, okay? All right, so I just want to show this, and I think it's the only time we'll do this in this course, but it's to show this. And so the next 3 slides, you don't have to learn about them, but just to see it once, okay? And what it's going to be is our favorite topic under a scab is PDGF, pathway, and MYC and cyclins, right? Like that, okay? So just look at this. Are June and Phos required for wound healing? That's the prompt. Yes, Phos and June are activated and highly expressed in cells beneath the scab during the wound healing process. They're crucial for inflammatory processes and proliferative processes, and there's a lot of overlap under a wound of who's doing what, but when we would think of June and Phos, again, we're not learning this, they play a big role in the immune response that's got to be happening, right? So under every scab, you also have some bacteria got into the wound. And so who's going to fight them? Blood cells, white blood cells, and this is not a course in immunology, but those guys show up and they get active and they start to divide. They start to increase their population to fight the infection. So yeah, and there's overlap. June and Foss gets activated in other cells, but when we think of it, and again, we don't have to think of it, we'd be like, oh, right. There's got to be an immune response, and that includes cranking those, white blood cells, the immune system cells, through the cell cycle too, like that, And how does that, what's that pathway, well, it's not PDGF, it's some other regulatory signals that are involved in the immune system. And we aren't learning that in our course, it's not immunology. But just to know that and see that, okay? And then the next prompt is hey, is that beta-catenin required under the scab? And that's the one that works with TCF. Right? Yes, beta-catenin is activated under a scab during the wound healing process, specifically within the dermal tissue, and becomes activated in the wound bed and soon after skin injury to initiate proliferative phases of repair. So it's got a role. It's an overlapping role, but this is where you're like, wow, I got hurt. It wasn't a superficial cut. It was an injury that went pretty deep, and I got to get some really repair healing going on some deeper cells. And we mentioned that earlier with SRFs. SRF is really a big deal at wounds, and that works with the elk to get things activated. And there's a long list of these things because there's a lot of action that happens under a scab. You're like, let me know all about everything that happens under a scab. And so it says, role in wound healing, activated beta-catenin acts to increase the migration and proliferation of fibroblasts, which are responsible for generating new tissue and collagen close to the wound. And so if it's a deeper wound, et cetera, like that. But then look at this. In summary, the scab marks a site of high molecular activity, including the activation of beta-catenin, which is essential for repairing the damaged dermal layers. So it's essential for those deeper ones. And when you have a bad owie, sometimes you get scarring. And that's actually part of the healing process. And the scarring is going to make that tissue tougher. So the idea is, well, you got hurt on your elbow, and maybe the skin on your elbow should be a little bit tougher for whatever stuff you're dealing with, right? And so that means, that kind of repeated wound, and you'll get more scarring and tougher skin in that area. It might be on your foot. So that's what some of the stuff is going on in healing too. And that involves proliferation, yeah, but it also involves secreting more collagen to just making the skin tougher like that. So there's a lot of stuff happening under a wound. Sometimes it's not the only time you got to cut there, right? Sometimes it's a cut that's abrasive, right? So you're like, okay, you better build up some tougher cells there on the skin, and you do that, right? So again, we don't need to learn it, but just a little glimpse of that, right? That we could have shifted and focused, not so much on MIC and instead focused more on beta-catenin, like that, and it would tell us a good story. It just turns out those stories are just not as clean, where you can just go and then, right, RAS and this and that and so forth, like that. Yep, all right, but just to see that a little bit, and again, our big favorite is that, phosphorylated elk working with SRF to activate MIC and cyclins and so forth. And so again, we can tell a nice long story there, about all the steps to get the cells into S phase, okay? And our book shows that. We've showed that a couple times, that pathway like that. And it starts with a push the button on the extracellular signal. So that goes back to chapter 16 and learning about cell receptors and signal transduction pathway. And this is just one example of the cell cycle of the cellular response that can be made by these extracellular signals, okay? So awesome. So we want to stay there and keep talking about that some more, okay? All right. And so we have this thing where we're like, okay, so over at start, we got to have these CDKs activated, and there are three big stories. And one of them is cyclins. And when it's cyclins, it usually means got to make more. So it's going to be a genomic response. It's going to be something about the cyclin cis-regulatory region and some transcription factors like those we just talked about getting activated and pushing it forward. In other cases, there's a phosphate and a phosphorylation of these, and we're not going to learn anything about them Our book really doesn't talk hardly about them either. That's fine, because we don't want to make it confusing. You know, we want to keep learning, but we've got a good phosphorylation story over at the G2 to M DNA damage checkpoint. So that exists, but don't worry about it. Then we got to talk some today about these small inhibitory proteins that are like the school patrol officer. They can just make the cells stop and wait like that. And they can do it for a bunch of reasons. And one of the reasons is, oh, you've been told to go through START. Are you doing the right thing, you know? Are you even looking for kids crossing the street going to school like that? You know, it's just the same as a school patrol type situation. They're repressors, they're inhibitors, all right? And so, you know, we could write that out. We have the visual, but cyclins, phosphorylation, and then the binding of these inhibitory proteins. And we're going to learn about two. One is called P21 and P27. And just to mention this, so biochemists often will name a protein just based on its size, because they don't know what it does. And so there are a lot of proteins that are just called P, some number. And that just means a protein that is 21,000 Daltons. So that's why people call these, most people call them small inhibitory proteins, because they're all really kind of surprisingly small proteins that bind, and their job is to inhibit. They always just inhibit, okay? And so we want to dive into some of those, and the way we're going to do that is to kind of organize this. There are 5 examples of START regulation that we're going to talk about, and we've already talked a lot about transcription and things like cyclins. There would be other stories about transcription and gene expression like that. That means we have to talk about these 4. 2 of them are small inhibitory proteins. So those will be our favorite examples of small inhibitory proteins, P27 and P21. That's their name, and we'll need to know their name. Then retinoblastoma and P53 are going to be these 2 most important examples for us of examples of tumor suppressors. And the way to think of those is oncogenes are the gas pedal at START, and tumor suppressors are the brake. They're like, yeah, no, not a good thing. And see, we've got the small inhibitory proteins. They act like a brake. You know, they would be considered tumor suppressors, but we have others that are bigger regulatory proteins, and that's their role. The regulatory role of retinoblastoma and P53 is just like hold on, like that. Hold on, okay? And they play the role at START. And they play the role where I say, really typically the light turns green, but initially it's kind of flickering and faint, and then we're going to crank it up. So we got a little bit of that in here also, okay? So retinoblastoma, I should just write down that really often people call it RB. Just RB like that. And it's a tumor suppressor. And so this is what RB does, okay? So RB is a co-regulator. It's a transcription factor that's a co-regulator, and it's a co-repressor. So tumor suppressor, co-repressor. It's trying to keep something turned off. And if you said, I guess what it is, you'd say something involved in S phase, right? The green light just means S phase. So that the car doesn't roll forward, the cell cycle doesn't roll forward, it starts copying the DNA. And so this is retinoblastoma's biggest target, and it's called E2F. And E2F is a very satisfying transcription factor for us to learn about, because we're going to learn about the targets. What is E2F trying to activate? It's trying to get the cell to do. And it is S-phase stuff, right? So this is how it works. So see, normally RB is just repressing it, and then if it gets phosphorylated, it lets go. And who does that? And the answer is the activated cyclins. So this is part of the biochemistry of turning light green. So you say, what are the targets of the CDKs? When the light starts to flicker green, what are the targets that start to get affected? And the answer is, well, we got some activated CDK, and it's gonna phosphorylate retinoblastoma. and help us push through the light into S phase. So that's a satisfying example of a target for activated CDKs at start. Retinoblastoma, yeah, Chase?
What was the name of the overall complex that CDK and cyclin form?
So the overall complex that CDK and cyclin form at start, that we are where we are now, does not have a special name.
Oh.
The overall complex over at the G to the M has that MPF, maturation promoting factor name.
Okay.
But here, start, they just don't have any kind of fancy extra name. But there's retinoblastoma. You can do that. We know so much about gene expression. You're like, okay, I could draw that. Okay, like that. Okay. So then these E2F target genes, right? So what do they do? You know, I mean, these E2F transcription factors that retinoblastoma is blocking. What do they want to do, right? The E2F transcription factors are downstream effectors of the retinoblastoma protein pathway and are required for the timely regulation of numerous genes essential for DNA replication and cell cycle progression. Several laboratories have used genome-wide approaches like CHIP to discover novel target genes of E2F, leading to the identification of several hundred freaking genes, right? Like that. When I was in high school, my friend, when I was 14, my friend Mark, who had his permit, got a car that could go 160 miles an hour, like that. And on the country roads in Minnesota, Mark and I used to drive around. I was a year younger than everybody in my grade, and Mark had a big brother, Rick, who had already bought this car. It was a 66 Nova SS. And we would race in the country and just drive down the roads 160 miles an hour when I was 14. That's what this is. That's what E2F. E2F was in the car with Mark and me going, yeah, let's go. Hundreds of genes. And what do? DNA replication. What's one of the target genes? DNA polymerase. What's another? All the enzymes you need to synthesize AGCT, like that. You know, the targets, you start out being, you're just like, oh, duh, we need lots of that. We had to turn into a DNA synthesis machine, factory, right? Big chromosomes. And so hundreds of target genes involved not only in DNA replication and cell cycle progression, but also DNA damage repair. You probably make some mistakes when you copy all those stupid chromosomes. Apoptosis, if you screw up, I don't know, we should be careful. Differentiation, development, blah, blah, blah. So E2F is just this monster, and it's a gene family of, transcription factors. And retinoblastoma has got the brakes on them, like that. It's got, it's holding on to Mark's foot, right? It's like that. We're like, Mark, I think we should slow down, like that, right? So E2F is just a great example for us, and retinoblastoma is a great example, right? Like that, all right? So now, listen to this, right? So a tumor suppressor means, if you get a random mutation, that's probably going to break the, it's going to be a hypomorph, a loss of function. It's going to decrease the activity, right? And you can see right here, right? If you start decreasing just the overall action of retinoblastoma, you have cells that are leading into it, right? E2F is active, and they may have not gotten all the signals to go into S phase, but they're making tons of DNA polymerase, like that, and tons of AGCT. You want to know who also messes with this? Viruses. Because viruses just want to take over the control of the cell, and they want to copy themselves, like that, So, just a really satisfying situation here, kind of like a bottleneck situation here with E2F. And there's other stuff going on, but gosh, that's important. All right, so here's a personal example to think about. Long before I was here, my family and I lived in Seattle. And I have two daughters. They're in their early 30s, so this is a long time ago because they were like in kindergarten. We moved here when one of my daughters was in first grade and the other was a little bit older, like that. Yep. So little kids, two daughters, and we had these friends, kid friends, family. We didn't really know them, but they had play dates and stuff. And that family had 2 little girls. And again, it was long ago in Seattle. You're not going to know these people. But this was also, you know, the life of this family. So they, you know, little girls are really cute. And the younger girl, who was probably 5, had one of her eyes was just closed. And it was closed in a really kind of sweet way where it was just gently closed. You know, her other eye was open all the time and working and stuff, but this eye was just like it was asleep, you know? And I'm just not the person to ever, you know, say, well, what's going on, you know, with your daughter, you know? But the parents and the family was, you know, really, really sweet people. And you could see they were, you know, just trucking along. And, but anyway, at one point, the dad he knew I was, you know, a scientist at the university, biologist, stuff like that. And we were there just playing, and he just mentions to me, he says that, I don't even remember her name, but he says, she's RB carrier, like that, okay? So think about what that means, right? What does that mean? It means that we all inherited 2 RB genes, 2 RB alleles from our parents. Ours are probably pretty good. The implication there was she had inherited from one of her parents a defective RB. So she was, you know, had a good copy and a bad copy, like that. And if she got the other gene mutated, you know, in her lifetime, right, she would be in trouble because she would have lost that tumor suppressor action, like that, right? And so I think what was happening was this. The retinoblastoma is called that because it was first discovered with some kind of very rare eye tumors. And I think that her family carried this defective version of retinoblastoma and was carrying that gene in their family. So they were sensitive to this retinoblastoma cancer. And I think that the doctors found this in her and they removed her eye because it was starting to form a tumor. And if in her lifetime she lost the other one, you know, in that other eye, right, like that, she might have to have her other eye removed to protect her from the cancer. Now, she had a bigger sister. Was her big sister a carrier? We don't know, because it's a chance event, right? Her bigger sister might not have had either, might have gotten 2 good copies of her parents. Because one of the parents must have been heterozygote, right? Because if you lose them both, you're in pretty big trouble, right? Like that. So one of the parents is probably heterozygote carrier, and the other homozygote, you know, wild type normal, and, you know, randomly going to get one of those, and then randomly they're going to lose it, like that. So, woof, right? And the molecular mechanisms of cancer are always like that. You're just like, oof, right? Shoot. Yeah. So that's why it's called retinoblastoma. It's got a bigger role. You know, it's just that's where there must be kind of like an Achilles heel in the pathway in our eyes, you know, like that. Okay? So going on with that, right? So then a little bit more about the targets. E2O. Did I already read that? Yeah. Okay? So, you know, targets, right? Blah, 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 right? But for targets, we should be able to realize, I bet they got to make a lot of DNA polymerase, and they got to make a lot of enzymes for DNA synthesis and synthesizing the nucleotides and DNA repair, you know, just logical stuff that you would need. I bet they could bind upstream of that MCM gene. Got to make more of the MCM proteins, more you know, origins of replication, get those going, blah, like that, you know. So good stuff for us to learn about. Role in the cell cycle, I just highlighted one thing. Yeah, among E2F transcriptional target genes are cyclins, CDKs, checkpoint regulators, DNA repair, replication proteins, like that. So they say cyclins too, right? Because that's part of this, the light's going to get brighter, you know, we're going to crank it up, like that. And so, again, you know, they could have a situation where if a person had an RB carrier, already had lost one, maybe they're already starting to make too many cyclins because they got some activity of E2F, you know, and they're just leaning into the cell cycle in a way that's dangerous, like that. Yep. Okay. So amazing stuff. E2F. E2F is a group of genes that encodes a family of transcription factors in higher eukaryotes. So paralogs, you know, through gene duplication, because they got a lot of targets and probably somewhat slightly different in, you know, different cells, different times, like that. But, you know, it's a family of transcription factors. Looks like it has maybe 9 members, no big deal, but you know, it's a gene family because it's got a big role and slightly different roles. And so then, this is interesting, I picked it. So 3 of them are activators, the other family members are repressors. So complicated story, right? You're like, oh shoot, you know, we just were talking about the activators, but it turns out in the gene family, we get this situation, right? And you can think of it sort of like, oh, you know, did you know you need you know, separate, you got a lot of screwdrivers. Some of them are just for screwing the screws in, others are for screwing them out, you know, like, you know, they're all screwdrivers, but they can evolve to have different roles in this complicated process, right? So good stuff. All right, so retinoblastoma, tumor suppressor. And see, we've learned about oncogenes like MYC and RAS and so forth, or B. That's a really good example of a tumor suppressor. And you see its function, right? Its function is to try to repress E2F, trying to repress the transcription factors that lead to S phase. Keep us out of S phase. So just a really satisfying example, okay? Retinoblastoma, all right? So then, that's our first one. Yay, you know? That's something about regulation of the cell cycle, and it's about what does it mean, you know, to move it forward. So our second example, P27, is in a figure in the book, and it's a super simple example, like that, okay? So P27, you know, it's a different example, it's super simple. And you probably can't see this very well here, but P27 is a small inhibitory protein, and it's going to block. It's like the stool patrol. It's repressing and keeping things stopped. And then you might be able to see here that it's going to get degraded. So that's going to help things. And how does it get degraded? The answer is it gets phosphorylated. And who phosphorylates it? The answer is CDK. So this is just a little example of how the light turns bright green. You know, there's these, you know, all kinds of checks and balances, you know, and this is one where, yeah, you could start flickering and I think it's time to go. But there'll still be that P27 hanging out saying, you know, like that. And you're basically saying, just get out of the, you know, way. We're going to have a fully green light and we're going to move ahead. So that's it. That's P27. You know, it's an inhibitor. It gets phosphorylated by activated CDK, and we get this positive feedback loop of, you know, a stronger activation of the transition into S phase. Okay, and that's that one. Okay, then here's this one, P53. See, it's a pretty big protein. It's not a small inhibitory protein. Okay, so we just have to make sure we see. These have all the same kind of names, but it's just their sizes, and they're very different proteins. They're not related at all. It's just labeling them from their size, like that. And so P53 is a big protein, and what does P53 do? And we're going to see that the P53 story fits together with that one. So the last one are going to be kind of paired together, okay? So P53 was the second tumor repressor gene identified. The first one was retinoblastoma, like that. And so it's inactivated in many cancers. So lots of times cancers have a mutation where the P53 gene is inactivated. How often? Mutations in P53 play a role in about 50% of all cancer. It's a tumor suppressor. It's not causing the cancer. It's the brakes, and you're not using the brakes, right? So the oncogenes are like the gas pedal, and they're like, let's go. And we have these tumor suppressors that are like the brakes saying, hold on. And if you lose the brakes, well, you're leaning in toward going. And so that's one way that cancers progress, right? So they could have a mutation in an oncogene like MYC, and they start causing trouble and growing too much, but then they start picking up additional mutations and just making it worse, making it more of a, okay, this cancer is just growing, and nothing the body has is stopping it, like that, yep. So that's a good example of that idea, right? And right, the oncogenes, the tumor suppressor genes, when they get mutated, they're really complementary, right? The oncogenes say, let's go. and the tumor suppressors are no longer saying wait, like that. So P53, okay? And P53 is a great one because it's a transcription factor, like that. And it's regulated by damaged DNA. So P53 is this DNA damage checkpoint at start. So that's where we have to talk for a minute because students get really confused by this. And it turns out that at start, we're mostly thinking about the going forward into the cell cycle. And in that process, that includes a DNA damage checkpoint. That's why I like the school control example, because the big story is if the light's red or green. But then there's also this option of making the car stop. And one reason they'd be forced to stop is because there's DNA damage. So think about that, right? So that you're under a wound, right? And PGF is saying, let's go. MIC is activated, RAS is activated, right? Everything's healthy and they want to go forward. But the CN cells are sitting at start, and let's say there was some DNA damage. And so this would be saying, the light's green, but here comes P53 and making all the cars stop, like school patrol, right? So it is a stronger repressor than the light could ever be green. Doesn't matter how green the light is, you have a totally separate mechanism saying wait, all right? And how does it make them wait? It activates the synthesis of P21. So our last two are connected. P21 is a small inhibitory protein. It has a protein, it must have a P21 gene, and who's sitting in the cis regulatory region of that gene? P53 can be activated and make P21. And that's that, right? And that's why I say, oh, that, you know, that, sorry, these small inhibitory proteins that we're at, and they're like this school control where they can just see, I don't care whether you have enough cyclins or enough phosphorylation, you're stopping. And that's P53, and that's DNA damage checkpoint. And the challenge is to keep it understanding separate from the G2 to M. So remember the G2 to M DNA damage goes through CHEK1 and CDC25 and removing the inhibitory phosphate. That's the G2 to M transition. That's that DNA damage checkpoint. Now we're over at start and we're mostly thinking about the light. There's a DNA damage checkpoint there too, and it's a big deal. Because if you have DNA damage and you start copying your DNA, right? That is just bad, 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 because you're just gonna maintain that damage. You're not fixing it. And cancer cells are fine with that. They're like, we're just dividing, if we start screwing up, that's fine. And what happens is that's how you start picking up more mutations, right? We keep saying, oh, cancer is a multi-hit thing. You gotta have a problem with this, oh, and that, and that. How's that happen? Well, you start screwing up DNA repair like that. You just like shrug, right? Let's just go, like that. So that's what's going on here. That's the sensible logic of the P53, P21 relationship. So then here's this other thing, and I mentioned this briefly, just briefly, but our next chapter is about programmed cell death, partly like that, right? And we're not going to learn these names in detail, but they're part of that chapter. And so this is what is going on. So when there's DNA damage, P53 in the next chapter, that's going to be on our exam, we'll see it binds to the P21 cis regulatory region, and we make the small inhibitory proteins. So that's like the cell cycle is stopping. This also has as target genes that will cause the cell to die. And there's a built-in timer. So that at first, if there's DNA damage, at first P53 says, let's fix it. But if you don't fix it in a couple of hours, you're gonna die, like that. And it just shows the significance of that DNA damage checkpoint. Because again, If you start copying damaged DNA, it becomes just a permanent thing. You can't fix it once it's been copied, and because you're like, I don't know, it looks good now, like that. So this just emphasizes that, that we're not going to let you hang out here very long because there's something wrong, and we're just going to have you go through program cell death. And again, you'll see these are members of a gene family with a bunch of funny names and a complicated story, because some of them are activators of program cell death, some are repressors. So don't worry about that. Just look at this, that we see p53 having at least 2 categories of target genes. It's transcription factors that binds the cis-regulatory region of p21, making the cell stop in the start as it fixes DNA. And then it also sets up this timer where you're going to fix it, or you're going to undergo program cell death. Yep. So just a really fascinating story, and a really super famous. So our last 2 are stuck together like that. Both of them do more, but that's just a great example of p53 and p21 for us to learn about. So, just back to the summary for the book, right? It's inactivated in many cancers and half the cancers like that. And it's a tumor suppressor. So for our exam, we're learning 2 really famous tumor suppressors, retinoblastoma, p53, and we want to know what their biochemical mechanisms of action are. Both of them are transcription factors. One, a co-repressor, the other, a transcription activator. Retinoblastoma does not have a DNA binding site because it binds on the back of E2F. E2Fs have a DNA binding site and hundreds of target genes. P53 has a DNA binding site. It binds the enhancer in the cis-regulatory gene of P21. And so we could ask on the exam to explain that or draw it, right? Just label it and draw it. Draw what we've learned about an E21 gene. And you just draw, a gene, right? P21, transcription unit, promoter enhancer, and then on the enhancer, put P53 like that. P53 is going to activate that when it's activated, okay? And we'll learn a little bit more about that switch in the next chapter, okay? So P53 is commonly referred to as the guardian of the genome due to its activities directed at maintaining genomic stability through the repair of DNA damage and integration of cell birth death signaling pathway with DNA damage checkpoints. So it's just saying what we just said, like that combination of repressing the cell cycle until DNA is damaged, and if it doesn't get damaged and fixed pretty quickly, activating program cell death. So it actually is a cartoon character like that, the guardian of the genome. So, cute, but again, half of all cancers, so also really serious. Okay, so that's that. And with that, do we want to, I'm just going to skip those because they're in our next chapter, really. And that school patrol guy, that's what that is if you don't know what I'm talking about, that analogy. And then, we'll talk about that in the next chapter. And I just want to point this out, that our book puts up 4 stop signs, right? And so again, we learned that just briefly about the spindle assembly checkpoint, the end, right? We know a little fact about that. Then we learned a fair amount of the G2 to M DNA damage checkpoint. And now we're over here, and at the G1 to S transition, the start restriction point, there's the P53 based DNA damage checkpoint. So 3 different places in the cell cycle with 3 different kinds of checkpoints, like in the washing machine a little bit, like that, And the book also has this, within S phase there's another checkpoint. We haven't mentioned anything about that, and we're not going to. I don't think the book really dives into that at all either, like that, okay? So I think we're doing great. This starts going into some the next chapter, talking in more detail. Oh, no, it doesn't really, not quite yet. So I just want to show you this, that, right, we learned 2 small inhibitory proteins, P21 and P27. I just want to show you that the book lists several of them. P15, 15, 16, 18, 19, 21, 27, 57, like that. So there are several of them. They're not in a gene family, they're just different, and they all do the same thing, and we learned about 2. Great examples, of that. I think that's the same figure, just zoomed in a little bit. Yep. And another indication of, oh, it's a family. I think this is another table from your book. And you don't have to go home and memorize anything about the others. We're focused on, our 2 good examples. So that's an example from the book about how the book kind of dives into some stuff in more detail than we would want to. Okay, great. Because I think we're now, at the end of that chapter 17, and we can start talking about programmed cell death and this stem cells. Yeah? So do people have any questions about where we're at right now?
Yes, Professor. Can
Yeah.
you go how P53 binds to DNA?
How a P53 binds to DNA? Yeah, so it's actually described a little bit more in the next chapter, but let's just, dive into it now and what's going on with that. So it turns out that when there's DNA damage at start, right? That's the point of the cell cycle we're at. So when there's DNA damage at start, there's going to be a pathway, biochemical pathway, leading to activating P53. And so how does P53 get activated? And the answer is when it gets phosphorylated, it's active. And so there must be a kinase, and we're not going to learn the name of that kinase. There must be a kinase on this pathway. It gets activated, it starts phosphorylating P53, and when P53 is phosphorylated, it binds to targets like P21G. So, again, label all these parts, but that's P21, and that's the enhancer, and phosphorylated P53 is going to bind to it and activate it. So what do? We add that to our list of transcription factors. Remember, for the first exam and first exam material, who else gets activated? Well, SMADs do, STATs do, CREV does, ELK does, right? Lots of transcription factors got to get switched on and off. That's a big theme in our course. And this is just another one added to that very familiar list. P53 is always hanging out. It's the guardian of the genome. It's the school patrol officer, and they're standing over on the sidewalk, over on the side. They're there. School's in session. They're there, ready to work, right? And but then P53 gets phosphorylated, and P53 is like the parent standing to the school patrol. It's the parent. P53 says, get out there, P21, and stop the cars, stop the cell cycle, right? So there's the school patrol gene, and you've got to make it, got to activate it. So that's P53 in activating P21. So we can draw that up, we can explain it. Then, this is just sick, because here's this, it will make it black because it's all spooky and dark like that, because this is a programmed cell death gene. And again, we're not going to deal with title names of these, but it's programmed cell death gene. When it gets activated, things are going to go dark really fast, like that. And here's the deal. P53 binds to it, but the form that binds to this is called a hyperphosphorylated P53. So to bind to this target, it has to get phosphorylated several times, and that's just how you get the timer. Is you start out, and this kinase pretty quickly phosphorylates P53, and it starts activating P21. But if you let that kinase just hang out, the DNA is damaged, it's active, it just keeps adding phosphates to P53. And eventually, there are several phosphates on P53, and it starts binding to this. And so that gene starts getting activated, And that's going to be a gene that's going to stimulate programmed cell death. And you could do an experiment, right? You could do an experiment where you have some cells, and they're stopped at start, like that. And you expose them to some DNA damage, like some x-rays, and then you start watching, and you take like a timeline, taking out samples every hour or every 10 minutes, and initially you start to see, we were detecting P21 RNA, now we're detecting it. The P21 gene is getting activated, so we start detecting the P21 gene. And you're like, okay, well, do we detect any of that programmed cell death gene yet? No. After a couple more hours, and it's a few hours, now we start detecting also the PCD1, the PCD gene RNA. It's starting to get expressed. And that's what we mean when it gets expressed, right? It starts to make the RNA and the protein product like that to do the function, right? And so that's what it means, experimentally is, oh yeah, pretty quickly when there's DNA damage, you start to see accumulation of P21 RNA and protein, and it's going to say stop, don't go, no matter what you think about the, start, don't go. You don't get to go. Whatever, you could be under a scan, right? We got to heal this and get these, this elbow back in shape. If there's DNA damage, you're just going to delay that healing, like that. And if it's a lot of DNA damage and you don't get it fixed, after a few hours, those cells will just get wiped out. Yep. Yeah, so it's sad, it's always like, and again, this is hard, of course it's hard because there's just lots of all these examples and they get jumbled together and stuff. But when we look at individual ones, you're just like, dang it, yep. That's how it's going to happen, right? And so, we emphasize, right, you know what? When P53, the tumor suppressor, is mutated, it's a random mutation, probably going to have a loss of function, a hypomorphic allele, decrease in activity. And at first I emphasized, yeah, so that just means, sorry, I shouldn't say that for that example. I went down the pathway for that example, but I don't want to use it. So that, yeah, okay, anyway. I started kind of thinking and using that retinoblastoma, but we're talking about P53. So P53, and that's it. That's what we want to learn about it. And it actually comes up a little bit more in the cell cycle chapter. Yep. Other questions people have about anything at all? Yeah, Don Diego?
Can you also draw the cis-regulatory region of the drawing of the cis-regulatory region that you say it's going to be busy with MIG, ELK, SRF, June 4th, and TCF?
Yeah. So this is that drawing, and we actually did it last time, and it's a good drawing, but we did it quickly, and so it's good to just loop back and take a look at it. So we should be able to draw this, and remember that at start, these cyclins have letters, and this is a cyclin gene. It's not B, it's one of the others, right? So we're not talking about B, we're talking about other family members. We don't need to know the letters. So just calling it a cyclin gene, that's great, but be aware that it's not B. We're not talking about B, B's involved in G2 to M. So, cyclin gene, and what we've drawn here, that's transcription unit, coding region, you can label that up, promoter, enhancer. And our first and favorite enhancer is that there is an enhancer that binds the phosphorylated form of ELK. And we learned that it also is bound to this other transcription factor called SRF. And both of them work together to activate expression. So both of them are activators. Okay? And then we learned that, there's a totally different, I guess I should draw the enhancers all the same color because they're all just DNA. Oh, here's another totally different enhancer, and it actually binds to June and FOS. Like that, and so we could also get cyclins activated sometimes by June and FOS. And then we also said, but you know what, there's another enhancer, because sometimes we got a situation where it's actually this TCF repressing gene expression, and the cell cycle gets activated because we bind stabilized beta-catenin. And then we also said that, you know what, sometimes the big super spooky one comes and it's MIC. And sometimes it's MIC that binds and activates them. And so that, that's a lot of stuff going on, right? And, but we want to get at least one example of that to be like, okay, I could see that there can be cis-regulatory regions that have lots of regulation. Yeah, Chase?
That's what I was going to ask. Like, these are all in the cis-regulatory region.
Mm-hmm. Yeah, they're all enhancers. And again, through our course, we've been emphasizing one enhancer, right? Myogenin gets activated by MyoD, and that's true. But even myogenin almost certainly has additional enhancers to regulate it in some other settings, blah, 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 like that, But then there's almost certainly more of these on the cyclins, like that. And maybe they're not all on the same cyclin, but we're not going to worry about that. It's just there's this family of genes, and they got to have a cis-regulatory region, and these are going to activate them. So, maybe they're all on the cyclin D gene, but like that. But yeah, we should be able to draw that. And Don Diego, you also asked about MYC, and so the deal is MYC is one of those, and so it's here's MYC, and what we learned is that ELK SRF, that's also bound here on MYC, and and I think that might be a reason why you asked this, is because sometimes the ELK story is, well, it's going to activate MYC, and then MYC's going to activate cyclins, and you're like, okay, all right. Other times, it is actually the ELK SRF that's going to activate the cyclins. So, and the book shows that too, right? Sometimes the pathway to the cyclins are a little bit complicated with an extra step, but it's because you're going to use another transcription factor like that. So that's why I think it can get a little bit confusing, because MYC also probably has a complicated cis-regulatory region, like that, but we're especially focused on that part of it, and then over here we're focused on all those, yeah? Yeah, and drawing them out, sometimes that's just, some drawing is worth 1,000 words, right? A picture worth 1,000 words. Sometimes it's just good, just be like, I got that, because I got it visually. And I could explain it, but a lot of times we can put together a better labeled, simple, non-artistic, we can put together a better labeled drawing than we can actually explain something, Somebody asks you, how do you get to the mall? I say, well, I might want to show you by drawing a map, Those could be very helpful, right? Instead of just being like, well, you go to the McDonald's, and like half-mile past McDonald's, right, by the old red barn, there's a fork, and you go to the right, like that. So a lot of times those are just helpful, both in explaining stuff, but also in holding that knowledge. So it's again, some of this is about learning, like that. Okay? Other questions people have, and it could be anything at all.
So can you come back in that image, please?
Yeah.
So the first exam you say describe, like the question you asked was describe, and then I remember I drove thinking about describing with an image. So can you explain what do you mean by describe?
So that means words,
okay.
Right? So this could be a drawing like that, and they do say draw and label, or you could say describe, and describe means you use words. So say this cyclin gene, every gene has two parts, transcription unit, cis-regulatory region, and the cis-regulatory region regulates where, when, and how much of the gene to express. And the most important regulators of the cis-regulatory region are transcription factors. That's where they bind. The cis-regulatory region doesn't change, it's the transcription factors, and so we're really interested in the switches, right? What makes that transcription factor do its job? So, you could have, it's there in the cell, but how does it get to do its job? And so, we have examples like that. Oh, it's got to get phosphorylated, or it's got to get stabilized, like that. And so that's the describe. And what I say is on an exam, if I say describe, you can use a drawing, labeled drawing also, because they're all, that's a very good way to demonstrate your learning. But I also want to have some words like that, because again, sometimes the person can draw it out, but they actually don't get it. They're like, oh, I don't know, it's the picture, don't you get it? Like that. So sometimes it's good, because the description ends up being the kind of cause and effect, like this does this and that, and kind of a timeline, whereas a drawing is always just a snapshot, or typically like that. So yeah, that's that. And so yeah, we want to pay attention to that on the exams. And it's not like I'm like a super fussy person, but it's just, yeah, you know, I really want to find out if the person can explain it with the words. Yep. Other questions people have? And we're doing fine. We have a couple more minutes. And so what's going ahead? Oh, yeah.
What is TCF again?
So TCF came up when we talked briefly about beta-catenin. In chapter 16, we had these different receptors. And we started out with the nuclear receptors and ester receptor. Nuclear receptors, oh, they're in the nucleus. They're transcription factors. So those are transcription factors that get regulated by ligands. And the ligand, the hormone just comes right into the cell. Then the rest of the receptors are all on the cell surface. And that's where we bring in this thing where you say, so the signaling molecule is going to bind on the cell surface. It could be a hormone or a local signaling factor, going to activate a receptor, and then there's going to be a signal transduction pathway, right, like that. And so for those, in chapter 16, we said, well, there are a couple of really big classes of those, categories of those. We got these 58 receptor tyrosine kinases that fit there, like that. And then we have hundreds of these G protein coupled receptors. And then the last part of the chapter says, here's some other signaling pathways that are really famous. And so we learned a little bit about SMAD and JAK-STAT and Notch, and we didn't learn about it, but TNF. And then one of them was this thing, the Wnt signaling pathway. And in the Wnt signaling pathway, the signaling factor is Wnt, binds to receptor frizzled, and the signal transduction pathway leads to the stabilization of beta-catenin. And beta-catenin is a transcription factor, but it's a co-regulator. It does not have its own DNA binding domain. It's going to bind on another transcription factor. And the transcription factor it binds on is TCF. So TCF is already there in the absence of Wnt. The TCF is bound to Wnt target genes, and it's repressing them. So it's a nice molecular switch because we're not seeing any gene expression. And then when Wnt shows up, that leads to the stabilization of beta-catenin, and that stable beta-catenin binds to TCF, overcomes the repression, and is actually an activator. So we get a big switch in those target genes. Yep. Other questions people have? So let me just show you one slide so you know we can prove we're all leaning into that next chapter. And again, the next chapter is about programmed cell death and stem cells. And we got that assignment coming up for Tuesday. I should mention the stem cell assignment video is pretty long. It's almost an hour long because I talk a lot about it because I really package really all the stuff in there. So just be aware of that. And then just to show you this first slide. So there's a really good chance that you've heard the word apoptosis as a kind of programmed cell death. And apoptosis is where a cell decides to die like that. And so it's dying not because of a lack of oxygen. It's not dying because it's sick. It's not dying because it's old. It's not dying because of poor nutrition. It's dying because it's gotten a biochemical signal and it's going to go through programmed cell death. And sometimes that signal is internal, like we just learned about. Sometimes the cell is just like going, whoops, I'm not getting the DNA fixed, and I'm going to have an internal decision to just wipe myself out, and that's apoptosis. Sometimes the signal comes from somewhere else. So like during our normal development, there's lots of remodeling that occurs, and I don't mean chromatin remodeling, I mean like a whole bunch of cells will die like that. And that will often be by an external signal saying, yep, you guys all need to get wiped out. You're done with your job. That's apoptosis. It turns out there's another, and our focus in this chapter is especially apoptosis. That's the one that's usually the cell is failing to die, right, like that. It's failing to die, and it'll go through apoptosis. There's another one that's another form of programmed cell death that's called autophagy, and it's actually described elsewhere in our book, and we probably won't read that chapter, but I just want you to be aware of that. So that, because that's why we call this programmed cell death, PCD, rather than just apoptosis all the time, is because there are actually two major forms. We'll especially talk about apoptosis. The only time autophagy will come up is I've just got a couple of slides from elsewhere to give you that as an example to compare and contrast with apoptosis. So we got to stop here. and we'll roll on next week. And I'll just say, we just have actually 3 class meetings before our next exam. So next week, we'll cover the apoptosis pretty easily, and then we'll go into chapter 19, which is adding on stuff. It's about cancer biology like that. And so we'll be in good shape. After spring break, we'll have a class on Tuesday where we'll get everything set, and then we'll have the exam on